So I'll give you a bit of an introduction about the results so that I can contextualise them properly. So as I said, I'm looking at results of the Abiraterone Phase III trial in STAMPEDE, and that recruited almost 2,000 patients between 2012 and 2013. The trial was closed to follow-up late last year, and the analyses that I'm looking at were from data that were cleaned and locked about 2 months ago. We collected blocks from patients enrolled in this trial, and with our collaboration with Veracyte, we were able to derive transcriptomes and transcriptomic signatures from diagnostic tissue from these patients. We had several objectives in this study. We wanted to test the prognostic effect of the Decipher genomic classifier in metastatic and non-metastatic patients separately. We wanted to see whether we could find an interaction between four transcriptomic signatures: the ARA signature, which is related to androgen receptor signaling, the PAM50 signature, which is a breast cancer signature, and also a recently derived signature of prostate subtyping, the PSC, and also the Decipher genomic classifier. So we had four signatures we wanted to test for interaction and one that we wanted to test for prognostic association. What we identified was that the effect of abiraterone was consistent, and the p-value for interaction with treatment was insignificant. So effectively, abiraterone works well for everybody. However, since these outcomes are variable, when we looked at the prognostic associations, and in this context it's looking at the Decipher genomic classifier, when we split patients, metastatic and non-metastatic, according to Decipher genomic classifier median score, which in our study was 0.77, what we found was that patients with a higher Decipher score, regardless of treatment with abiraterone or ADT, did worse than patients who had a Decipher score above the median, and this was a significant prognostic finding. So what we can see here is that Decipher is helping us to stratify patients and potentially to help clinicians and physicians make decisions about treatment. What we found was that the relative benefit of abiraterone was consistent. However, the absolute benefit is different. When we looked at 3 different time points in both metastatic and non-metastatic patients, where we split patients by their Decipher genomic classifier score, we found that the absolute benefit in metastatic patients with a higher Decipher genomic classifier score, there was a 32% increase in absolute benefit for patients treated with abiraterone. In non-metastatic patients, there was an increase of 23% of absolute benefit at 96 months. So it's these kinds of differences that we want to investigate further, and that we hope physicians will be able to use in the future to be able to make treatment decisions.